Hello students, last class you have studied about sexual reproduction flowering plants, that is the male gametophyte, that is the androecium. So in that you have studied what is microsporangium and microsporogenesis. So today let us see the female reproductive part, that is the gynoecium. Female reproductive part, it have a single pistil, that is monocarpillary, or more than one pistil, that is multicarpillary. In picture you can see the hibiscus pistil, it is a monocarpillary, whereas the remaining two, that is papaver and mechila, it is a multicarpillary. In multicarpillary, the pistils may be fused together or free. If it is fused, then it is syncarpous. If it is free, then it is apocarpous. About types of carpillary you have studied in your first period class now. Recall that. Each pistil has three parts. First one is the stigma. This is landing platform for pollen grains. So in the region of stigma, the pollen grains. So pollen grains, it is nothing but a androecium part, right? It is one of the part of the male reproductive system. So pollen grains, it will stand on that stigma. It is a landing platform. Style, it is the elongated slender part beneath the stigma. The elongated one from the stigma is the style. Next is the ovary. It is a basal bulge part. So ovary region, it is little fatter one, bulged one, enlarged one. Inside the ovary is the ovarian cavity, the locule, in which placenta is located. In the picture you can see now that ovary and ovule, that arrangement is nothing but the locule. The empty space is the ovarian cavity, that is a locule. Arising from the placenta are the ovules. So from this placenta region, the ovules are arranged. Those ovules are nothing but the microsporangia. The number of ovules in the ovary may be 1. Like wheat, paddy, mango, they are all having only 1 ovule. Whereas too many in papaya, watermelon, orchids. Now let us see the structure of megasporangium. So megasporangium is nothing but the ovule. So inside the ovule, what is there? It's very important. Observe the picture clearly. Ovule is attached to the placenta by a stalk, the funicle. So for this ovary is attached by this funicle. And junction between the body of the ovule and the funicle is called helium. The one region which attaches this ovule and the funicle, no, that is the helium. Each ovule has 1 or 2 protective envelopes, that is the integuments, except at the tip where the small opening micropyle is present. So there are 2 layers of integument, that is outer integument and inner integument, that is like layers there. But in the region of micropyle, the top one, there it is not present, there it is an opening there. So opposite to the micropolar end is the chalaza, there is a basal part. So many students, what they do, they mix up this micropolar end and the chalazal end. So don't get confused. Chalaza is the basal part, micropylla is the opening side. Okay. Enclosed within the integuments, there is a mass of cells called nucleus. So after the integuments, there are n number of cells. That region is called nucleus. It contains is the food materials. Inside the nucleus is the embryo sac. You are having integuments, two integuments, then the nucleus. Inside the nucleus you are having the embryo sac. An ovule generally has a single embryo sac formed from a megaspore. So there will be only one embryo sac. Microsporogenesis. So this megasporogenesis. This megasporogenesis, it takes place inside the ovule. See, what is megasporogenesis? It is a formation of megaspores from the megaspore mother cell. Ovules generally differentiate a single megaspore mother cell in the micropylar region of the nucleus. You can see the micropylar and the nucleus, and you are having a megaspore mother cell. There is only one megaspore mother cell is present. It is a large cell containing dense cytoplasm. It is having very thick cytoplasm and the prominent nucleus. One clean round nucleus is present. So megaspore mother cell undergoes meiosis to produce 4 megaspores. You know, when meiotic division means it will result in 4 cells. So this megaspore mother cell, what happens? It undergoes meiosis. Then you will be getting 4 megaspores. First will be the dyad, then comes the dyad means 2, tetrad means 4. So now we are having 4 megaspores. Formation of female gametophyte. In majority of flowering plants, one megaspore is functional while the other 3 degenerate. In the previous slide, you came to know that there are 4 megaspores. Now in that 4 megaspores, what happens? The 3 will not function. Only 1 will function. What happens? The remaining 3 gets degenerated. It is like it is getting eliminated. See, in this picture it's very clear. Functional megaspore you are having 1. The remaining 3 is getting degenerate. So degenerating megaspores. So this functional megaspore, it develops into the female gametophyte. The embryo sac formation from a single megaspore is called monosporic development. Means now you are having only one megaspore, right? This megaspore, it will develop into a embryo sac. Only one. That is the reason it is monosporic development. Mono means 1, right? So it is monosporic development. The nucleus of functional megaspore divides mitotically to form 2 nuclei. Now there were only 1. That is the monosporic development. So only 1 megaspore was present. Now what happens? It undergoes mitosis and you'll be getting 2 nuclei. They move into opposite poles forming 2 nucleate embryo sac. The nuclei again divide 2 times forming 4 nucleate and 8 nucleate stages of the embryo sac. First it will get 2 will get divided. Again that 2 again when they divide, what happens? Total it becomes 4. That 4 again when they divide, what happens? It becomes 8. There is the 8 nucleate stages of the embryo sac. These divisions are free nuclei. That is, nuclear divisions are not followed immediately by the cell wall formation. It's not like when they get divided, when the nucleus is divided, this start forming a new cell. It's not that way. These are free, so cell wall formation doesn't take immediately. After 8 nucleate stage, cell walls are laid down, leading to the organization of typical gametophyte. Now see, that time though it is free nuclear, cell wall formation did not take place immediately. Now what happens? Cell wall formation is taking place of the typical female gametophyte. 6 of the 8 nuclei are surrounded by cell walls and organized into cells. So there were 8. Out of 8, what happens? 6 are having the cell walls, which will be organized into the cells. The remaining 2 nuclei, that is the polar nuclei, are situated below the egg apparatus, the large central cell. The two which is remaining, that is the two polar nuclei. A typical mature embryo sac is 8 nucleate and 7 celled. 8 nucleus means it is understood it's having 8 nucleus. 7 celled means now cell wall formation, microphylla, and you are having 3 cells. Top in antipodal region you are having 3 cells. So there's 6. Overall you are having 1. So that's how it is 7 celled and 8 nucleate. 3 cells are grouped at the microphylla end and form the egg apparatus. The 3 cells which are found at the microphylla end is called the egg apparatus. It consists of 2 synergids and 1 egg cell. Synergids have special cellular thickenings at the microphylla tip called filiform apparatus. In that picture it is marked filiform apparatus. They are like finger-like projections. Okay. It helps to guide the pollen tubes into the synergid. That we will be studying in next class. Means the pollen grain will enter the embryo sac with the help of this filiform apparatus. And the 3 cells at the chelazal end are called the antipodals. The large central cell has a 2 polar nuclei. So the overall, see how it looks. They undergone meiosis and the 4 cells were formed. In them what happens? Only 1 spore remains. The remaining 3 gets vanished. Means they get degenerated. Then what happens? The single functional megaspore, it undergoes mitotic division. It will be 2 cell stage. That 2 cell again it undergoes mitotic division. You will be getting 4. And that 4 it undergoes mitotic division. You will be getting 8. So here what happens? The 3 out of 8, 3 remains in the chelazal region. 3 move towards the microphylla end. The chelazal region is the antipodals and the microphylla end is the egg apparatus region. The remaining 2 nuclei, they remain in the center. So this is how the mature female gametophyte process takes place. So related to this microsporogenesis, megasporogenesis, I will explain it to you in the next class. The overall, I will explain to you with the video. So important questions in this. What is gynoecium? And explain the structure of megasporangium. That is the ovule that integuments, helium. So those regions along with the diagram. And third one is explain the formation of female gametophyte. So now we have seen that meiosis is taking place, mitosis is taking place. So that you have to explain. Thank you.